Drop a tablet into a glass of water, and within seconds it begins to disintegrate. As consumers, we expect this reaction. We implicitly trust that a manufactured chemical compound will behave exactly the same way inside our bodies, dose after dose, year after year. But in reality, that baseline consistency is entirely unnatural. It is a highly engineered state maintained by an uncompromising global regulatory framework. Even if a laboratory perfectly synthesizes the correct active pharmaceutical ingredient, the final drug product can still catastrophically fail in a patient's bloodstream, simply because the physical chemistry of the pill is flawed. What we perceive as absolute safety is actually an invisible architecture of tests, limits, and controls designed specifically to prevent systemic public health failures. The blueprint for this architecture is a document known as the ICH Q6A Guideline. It is the harmonized global rulebook used by the United States, Europe, and Japan to establish quality standards for all new chemical entities. In regulatory terms, a specification is a strict, legally binding list. It defines the exact analytical test procedures to be run and the numerical acceptance criteria a drug must meet to be considered safe. The guideline divides these requirements into two distinct categories: universal tests, which apply to every single new drug, and specific tests, which vary based on the drug's intended physical form and delivery mechanism. The goal of these specifications is not to achieve full molecular characterization of the drug. Rather, they serve as the terminal validation step for a manufacturing process that is already deeply understood. ICH Q6A acts as the algorithmic logic that translates theoretical physical chemistry into a guaranteed clinical reality. Every new drug substance must pass 3 universal tests: a description of its physical state, a specific identification test, and an assay to confirm its exact strength. But it is the 4th universal requirement, impurity testing, that presents a massive statistical challenge. Manufacturers must figure out how to extrapolate safe impurity limits from early, small-scale clinical batches to massive commercial-scale production. This Q6A flowchart traces the logic for specific impurity limits. First, manufacturers determine mean impurity levels across development batches and calculate an upper confidence limit. Following the decision tree, they must distinguish if this impurity is also an active degradation product, meaning its volume increases over time. To lock in the upper confidence limit mathematically, the regulation strictly defines it. It is exactly 3 times the standard deviation of the batch analysis data, plotted against the mean. This second decision tree illustrates how degradation products are managed in the final manufactured pill. Regulators require manufacturers to estimate the maximum likely level of that degradation product at the very end of its shelf life, using data extrapolated from accelerated, high-stress stability studies. Acceptance criteria for impurities are never arbitrary numbers. They are mathematically derived predictions of long-term safety, weighed against strict toxicological thresholds. When dealing with a solid oral dosage form, the chemistry is irrelevant if the pill cannot physically break down in the gut. Testing the mechanical release of the drug is a critical specific test. This decision tree determines correct drug release acceptance criteria, starting by checking if the drug is highly soluble across the physiological pH range. If the dosage form dissolves rapidly, the guideline offers a specific regulatory alternative. A simple, time-capped physical disintegration test can replace complex dissolution testing. However, looking at this section of the flowchart for immediate-release drugs, where dissolution strictly dictates bioavailability, the requirements are vastly different. Manufacturers must adopt specific test conditions and acceptance criteria, explicitly designed to catch and fail any batches that would perform unacceptably in a human patient. For extended-release products, the complexity scales up again. As this diamond shows, establishing an acceptance range requires a proven in vitro-in vivo correlation, meaning laboratory dissolution results must reliably map to human clinical data. By tightly engineering physical disintegration or dissolution tests, the specification provides a highly controlled proxy for how effectively that drug will enter a patient's bloodstream. Even if a drug has the perfect chemical formula, it can take the wrong physical shape. Molecular lattices can shift into different, tighter geometric patterns. These shifting crystal forms are known as polymorphs. Because the molecules are packed differently, polymorphism can drastically alter a drug's solubility, its melting point, and its baseline chemical stability. This decision tree mandates an active screen for polymorphism during drug substance development. If different forms can be created, they must be fully characterized to see if they possess different physiological properties. But controlling the raw drug powder is insufficient. This sequence from the guideline demands active monitoring of the polymorph ratio during the stability testing of the final solid drug product, checking if the crystal structure silently changes while sitting in the package. Active physical surveillance prevents a chemically pure drug from spontaneously shifting into a form that is physiologically useless or dangerously toxic on the pharmacy shelf. A similar threat occurs with chiral molecules, compounds sharing the exact same atoms but existing as non-superimposable mirror images, triggering drastically different biological effects. Standard analytical assays cannot distinguish between left-handed and right-handed molecules. If a manufacturer uses an achiral assay, they are completely blind to the presence of the wrong, potentially toxic, enantiomer. This flowchart details requirements for single enantiomer chiral drugs, mandating stereospecific testing. However, footnote 4 provides a narrow exception. A manufacturer can use a narrow achiral assay for strength only if combined with a rigorously validated procedure that explicitly measures and controls the opposite enantiomer. Without stereospecific testing, regulators cannot legally guarantee which version of a chiral drug is actually binding to receptors inside the patient's body. Testing a finished batch of pills at the end of the line is ultimately just one piece of a total control strategy. True pharmaceutical quality is built on a foundation of good manufacturing practices, in-process testing, and deep process validation. To ensure that control holds over time, regulatory release criteria are often tighter than shelf life criteria. A manufacturer might hold a drug to a strict 98 to 102% purity band before it leaves the factory, guaranteeing it will remain within the wider 95 to 105% legal bracket for its entire shelf life. When the manufacturing process is perfectly understood, Q6A allows for parametric release, an operational alternative that eliminates routine end product release testing entirely. For terminally sterilized drugs, instead of pulling finished vials to test for bacteria, the release of the batch is based on validated physical data. An industrial autoclave records precise pressure and temperature metrics throughout the sterilization phase. Continuously measuring those physical parameters across the entire cycle is vastly more reliable in predicting sterility for the whole batch than randomly destroying a single sample to test it in a lab. The deepest levels of safety are engineered directly into the manufacturing environment itself, rather than merely verified at the exit door. When a patient picks up a prescription, they are holding the physical manifestation of a massive data structure. Specifications are the mathematical translation of chemical mechanics, converting theoretical science into guaranteed, repeatable safety and efficacy. Maintaining this uncompromising global standard requires intense, hidden labor from pharmaceutical and regulatory scientists, running thousands of analytical tests before a product ever reaches a pharmacy. The public's trust in modern medicine is not an assumed given. It is actively earned every single day by the stringent logic laid out in guidelines like ICH Q6A. Every time a pill works exactly as expected, it is a quiet triumph of analytical chemistry over biological chaos.